This is a depiction of a competitive immunoassay in which the antigen has been bound to an enzyme. The enzyme catalyzes a particular chemical reaction in which a chromogen, which is added later, then changes color, and this color then can be analyzed. In this first sequence, there is only antigen with bound enzyme, and so therefore the amount of red pigmented product produced is the maximum amount possible. In subsequent runs, in addition to the antigen which has been bound to the enzyme, unlabeled antigen, which is not bound to the enzyme, is also added. And this then competes with the bound antigen for the binding sites on the antibodies. After the unbound antigen is washed away, this applies to both the bound and unbound antigen, then the amount of antigen with its enzyme bound, which is attached to the antibody, this will determine the amount of chromogen which is converted into the pigmented product. And thus, a series of standards can be developed which one can compare an unknown to. So not only would one be able to identify the presence of unlabeled antigen, but also the amount of this unlabeled antigen, because the greater the amount of the unlabeled antigen, the more it would compete with the antibody binding sites, and thus the less of the antigen with the enzyme attached would be able to bind the antibody. Thus, the greater the amount of antigen in the sample, the unbound antigen, the less chromogen will be converted into a pigmented molecule, and the lighter the color.